All right, perfect. So, good evening, guys. Today I'll be talking about the tumors of hematopoietic and lymphatic tissues. Now, this question belongs to the general pathology questions, and therefore, the way you would go about this is that you actually study the organ-specific questions of hematology, and then you can get back to this once you're done with the pathology of the blood and the lymphoid tissues, and then you can, just look into this question. So, because that's the best way to do it and the most efficient way to do it, I'm just going to run through the things that you would have to include for this, and I won't go into the details of hematopoietic and lymphatic tissue tumors because you're going to cover the same thing even in organ systems. So there'll be a dedicated video for that where I'll explain in greater depth. Now, for this question in general pathology, the best thing to do is you just talk about, you give a basic introduction about the tumors of hematopoietic and lymphatic tissues. You give the classification and a very brief explanation, maybe just one or two sentences, or maybe something extra that you can add on, which is very important to explain. Just explain that for the subtypes of the classification. And then it's very important that we talk about the signs and symptoms of the leukemias and lymphomas. On top of this, you can also add the differences. I will type it over here. Leukemia and lymphomas. So you can just mention this as well. I think it's a very good way of understanding the differences and knowing this topic properly. So let's just get on with it. Now, when you look at the tumors of hematopoietic and lymphoid tissues, it will affect the blood, the bone marrow, the lymph and lymphatic system. Why? It's because the immune system and the circulatory system are connected functionally and even structurally. Now, we know that there are two basic lines, cell lines for hematology. You have myeloid and lymphoid cell lines. Myeloids are responsible in producing granulocytes, such as neutrophils, basophils, eosinophils, and then you have erythrocytes, thrombocytes, macrophages, and mast cells. Whereas lymphoid is lymphocytes. Now, in lymphocytes, you have B, you have T, you have natural killer, also known as NK cells. And of course, you have plasma cells, which are basically the mature B lymphocytes. So you have to get this fact very clear. You have the myeloid line and you have the lymphoid line. Now, based on this, we have further classification, and you can understand the whole topic. Before I get on with the classification, it's important to note that when you go to the notes, especially in organ systems, you're going to come across a lot of chromosomal translocations. For example, translocation 1712, etc. So it's important that you know these. So pay attention to chromosomal translocations because they're a common cause of these diseases. So let's continue. So this is just a picture. You have the multipotent hematopoietic stem cell, which will differentiate into myeloid and lymphoid lines. Myeloid will give rise to your thrombocytes, BEN, that's Ben, and monocytes, which can further, I would say, mature into macrophages. For the lymphoid line, you have natural killer, you've got T lymphocyte and B lymphocyte. The mature version of B lymphocytes or the activated versions are plasma cells, which are capable of secreting antibodies. Now, how do you diagnose these tumors? This is just a basic explanation. There's definitely more to this. So usually we always start off with a complete blood count, and we also look at a differential count of the white blood cells, where we understand how much percentage of each of these cells are present within the blood. And obviously, one of the better signs for these tumors is lymphadenopathy, which is LAD here in short. And when we suspect some sort of a tumor with the hematopoietic stem cells, or in hematology in general, we often take a biopsy from the lymph nodes. Now, historically, when you look at the classification was based on whether this malignancy is there in the blood, and that is called leukemia. So you have emia, which denotes for blood. or you have malignancies affecting the lymph nodes, which is then called lymphomas. So understand that leukemia means there is a neoplastic proliferation of the hematopoietic marrow cells, and this is getting into the blood. So you have the tumor, or you have this neoplasm existing in the circulatory system, in the blood. Whereas in lymphomas, you have this neoplastic proliferation of immune cells, but they are occurring in the lymph nodes, and then they might spread to the blood and vice versa. Even from the blood, the neoplasm can shift into the lymph nodes. And that's why later on in the video and also in the organ system questions, I talk about how you have transition from lymphoma to leukemia and leukemia to lymphoma. All right. So in lymphoma, again, you have lymphoid tissue. That is the main origin of neoplasm. In leukemias, you have the bone marrow or the blood in general, where the origin of the neoplasm is. Now, obviously, blood is a type of connective tissue. These tumors are usually a mesenchymal type of neoplasm. So just know this as well. You should know this. It's a pretty important detail. Now, let's go for the classification. So again, number one, know what is myeloid, know what is lymphoid, know the difference between the two. Myeloid stands for uh, the granulocytes, erythrocytes, thrombocytes, and did I miss anything else? Um, yeah, macrophages and mast cells. Okay. Lymphoid is B cells, B lymphocytes, T lymphocytes, and NK cells. Now, if you're going according to the lineage, you differentiate as myeloid and lymphoid. And under lymphoid, you have lymphomas, okay, which is basically what I mentioned earlier. And again, like I mentioned earlier, you have it either arising in lymph nodes or in extranodal tissues. Essentially, extranodal tissues, I mean tissues other than lymph nodes, where you have this aggregation of lymphocytes. For example, if you consider the intestines and if you consider GIT in general, right? Especially in the small intestine, you have so many lymphoid follicles. And even if you consider the pharyngeal mucosa, you have multiple follicles, you have tonsils, right? So these are the differences. Um, Now, again, these lymphomas can transform into leukemias, and some lymphomas have actually leukemic presentations. So, other than myeloid and lymphoid, we also talk about histiocytic or dendritic cells and mast cells. Now, about these, again, you can just go to the organ system questions, and that's where we'll cover most of it. Once you do organ systems, you just come back to this question, and this question you can finish it in like 10 minutes or even 5 minutes for that matter. Now, if you go, I'm just going to skip this because this is typically what you'd have to mention for every single subtype when you're going into depth. Now, if you look at the classification of these tumors, according to WHO 2001, uh, myeloid neoplasms, which I mentioned over here, you have three main types that I want you to remember. There's chronic myeloproliferative disorders, okay? You can, because it's a, so myeloid lines typically affect are occurring in leukemic situation. So you can just talk about chronic myeloid leukemia, okay, CML. Uh, and since over this, the definition is that uh, the cells, the hematopoietic stem cells, are going to undergo this terminal differentiation. However, the manner in which they undergo this terminal differentiation is dysregulated. Okay, so they are not perfect neutrophils. They need not be perfect erythrocytes. There will be imperfections, and as a result, there will be a neoplasm arising from it. So, um, there will be an increase in one or more of the formed elements. Very important point to notice, if you look at this picture, uh, okay, if you look at this picture, if there is some dysregulation in this specific cell, you can be quite sure that all of these cells are going to be affected in some or the other way, right? Similarly, if let us say that there is a dysregulation that starts from the common myeloid progenitor, you can expect some sort of neoplasm to be arising within these cells, but maybe not so much in this, right? So understand that concept. that the more pluripotent or the more primitive the cell is, the greater chance that the entire line will have a neoplasm or some sort of a dysregulated growth. Now, okay, we'll do this. Yeah. So that's why if you have any dysregulation in the initial cells, in the pluripotent cells, or in the progenitor cells, there can be an increase in one or more of the formed elements. So red blood cells, platelets, and or granulocytes can be affected. Now, under chronic myeloproliferative disorders, you're going to have chronic myeloid leukemia, polycythemia vera, which is essentially an increase in the RBCs, of course, with the dysregulation, primary myelofibrosis, which again, I'll come to it in greater detail in organ systems. You just have to know, you just have to get an outlook, a basic understanding of what this tumors are all about. And essential thrombocythemia, which in which you'll have thrombocytopenia and you'll have increase and a decrease as well. Now, you can also have chronic neutrophilic leukemia and chronic eosinophilic leukemia. Now you might ask the question, if this is a tumor and if you're going to have a neoplasm, you'd naturally expect a large amount of cells to be there. Then why is it that we say there can be cytopenias associated with these? And that's because we are saying cytopenias with relation to how efficient the cell is and how close it is to a perfect regulated cell. So there can be a situation where there'll be a lot of RBCs, but they're not perfect in carrying, in performing their duties, in performing their functions. And in that way, you're going to have a lot of these progenitor cells, which are sort of dysregulated, or you're going to have erythrocytes, which are formed from a very dysregulated manner, and therefore they're not exactly erythrocytes. And you're going to have a lot of those, and that will form the neoplasm. Whereas the actual erythrocytes, the normal ones, will be low in number. And that's why you could say that, okay, there can be erythrocytopenia, for example. Now, so that is basically chronic myeloproliferative disorders. You have a terminal differentiation occurring, but in a dysregulated manner. In myelodysplastic syndromes, what's happening is that there is a multipotent stem cell, okay, and this is going to undergo some transformation, some dysregulation, and it's not going to be the healthy one. That will form, that will replicate, and that will form a clonal progeny, right? Now, this clonal progeny retains its ability to differentiate into specific cells. So therefore, they can get differentiated, but again, the order in which they're getting differentiated is both ineffective and it is disordered. Okay, so here, for example, you can clearly see the cytopenias because, like I mentioned earlier, there will be a chance where you have a huge growth of all these clonal progeny. However, the effective cells and the perfectly healthy cells are very low in number, and that's why you can have cytopenias. Over here, you're going to have an increase in the number of cells. Okay, it's a proper increase. There can be stages of cytopenias, but that's maybe just transitional. But predominantly over here, you're going to have a myeloproliferative disorder, a proliferation, an increase in the number of formed elements. Okay, so here in myelodysplastic syndrome, again, like I mentioned here, you're going to have a cytopenias, but again, there can be stages of hypercellular marrow as well. And hypercellular is because you have a neoplastic proliferation of the clonal progeny, and that's why it's hypercellular. They can also be hypocellular when the resources in the bone marrow are exhausted, and therefore the cells are not being produced in efficiently. Now, uh, there will be changes in the morphology and maturation for sure because there's going to be a dysregulation in the growth, right? So you can call this as a dysmyelopoiesis. Poiesis means the growth and differentiation and maturation. So this means dysregulated. And of course, again, you'll have a peripheral blood cytopenia of either one cell or more cells. Um, Yep, and you will definitely have ineffective hematopoiesis. Again, this comes back to the fact that there will be an ineffective and disordered manner of differentiation. So, um, another point is look at this. The abnormal stem cell clone in the bone marrow is genetically unstable and prone, and they can lead into AML. A lot of these leukemias can go into AML, acute myeloid leukemia, and that's where there is more than 20% blasts. Blast means the primitive stages, right? Erythroblast, for example. So uh, there'll be an increase in these undifferentiated cells, and when it goes more than 20%, it's called AML, acute myeloid leukemia. This is very commonly found in chronic myeloid leukemia, where there can be a sudden change, and there all of a sudden there can be a sudden increase in the number of or increase in the turnover of these dysregulated cells, and as a result, the blasts, which are still primitive, will be more than 20% in the blood. So that in that situation, it's very dangerous. It's a potentially fatal situation. That's called acute myeloid leukemia. It's an acute stage, acute situation. Now, under MDS, myelodysplastic syndrome, the basic classification is, I think there are about five. So you have MDS with single lineage dysplasia, with multilineage, with excess blasts, with ring sideroblasts, with there's also something else unclassified, and chronic myelomonocytic leukemia. Now, again, you go back to the organ system question, learn about this in depth, then come back to this and just sort of summarize it. This question is nothing but a summary of the organ systems. And I think a lot of the other neoplastic questions are as well. Now, acute myeloid leukemia. So, you have the early stage of myeloid cell, which is mostly blasts. Uh, the blasts are prevented from further differentiating and maturing. So what happens? You'll have a buildup of blasts. When it goes more than 20% of the blood in the blood, acute myeloid leukemia. Okay, that's with relation to myeloid.